Okay, looking at glycolysis, the name literally means the splitting lysis of glucose, glycose. So glycolysis, the splitting of glucose. So it's the first stage of respiration. It takes place in the cytoplasm and it is involved in both aerobic and anaerobic respiration. We can, broadly speaking, divide the process of glycolysis into 4 stages. Phosphorylation, so the addition of phosphate. Lysis, so the splitting. Oxidation, the removal of hydrogens. And then ATP production. Okay, looking at the process of phosphorylation. Now, this process is a little bit like when you strike a match, you have to put energy in. So when you strike a match, you're putting energy in by rubbing the head of the match against a rough surface, so for friction, and that then allows there to be enough energy for there to be the burning of the matchstick. Well, this is essentially what's going on here. We're putting in some energy to then allow a whole load of energy to be released. Because glucose is an incredibly stable molecule. It's not just going to suddenly split itself into 2 just because of us wanting to get energy out of it. We need to give it a bit of a push. So that's where this phosphorylation happens. So what happens is that the ATP is broken down into ADP and P, and then that P will then join the glucose, initially to form something called glucose 6-phosphate, and then it happens again, and that forms something called glucose 1,6-phosphate, or more generically, hexose biphosphate. So what that tells us is that it's a 6-carbon sugar with 2 phosphate molecules, okay? And it's completely fine to use the generic terms like this, hexose biphosphate or that sort of thing. You will see some places it's written as glucose 1,6-phosphate. Sometimes it's called fructose biphosphate. There are lots of, because this process is inevitably rather more complex than what we're looking at. We're just looking at the basics. And so there are, I'm afraid, lots of possible names here. So that's the phosphorylation. And the purpose of that is to kind of give an energy boost, if you like. Once we've done that energy boost, then we can start to do the lysis. So by adding 2 phosphates to the glucose, you make it sufficiently unstable that it is able to split into 2 triose phosphates. So hexose biphosphate is 6 carbons, and triose phosphate, as its name says, triose 3, and it's still got a phosphate. That's 3 carbons, so we need 2 of them. So basically our 6-carbon sugar has now been broken down into 2 3-carbon sugars, and that's the splitting phase. In the next stage, which is oxidation, remember, oxidation is loss. So the triose phosphate is going to lose a hydrogen. So basically that hydrogen is going to come from the triose phosphate, and then it's going to be added to the NAD, which, if you remember, are our little shopping trolleys, our hydrogen carriers, and the NAD becomes reduced. So that's an example of a redox reaction just there. So the triose phosphate is oxidized, the NAD is reduced, and that's a way of us storing our NADs. Now, a little point to note here is that we started with 1 6-carbon sugar, and now we have 2 3-carbon sugars. So from here, everything happens twice. So because we've got 2 triose phosphates, we're going to end up with 2 reduced NADs, because each triose phosphate is now going to go through this whole process. After the oxidation, we then have ATP formation. Now, the vast majority of ATP that is formed in respiration is formed using a process called oxidative phosphorylation. So phosphorylation using oxygen, that we will discuss in great detail at a later date. But glycolysis, as you know, is the part of respiration that can also occur without oxygen. So this ATP formation is done through what we call substrate level phosphorylation. And you can see here that the triose phosphate, it's been oxidized. So here's its oxidation. And then here it's making 2 ATPs. Now, because we have 2 triose phosphates, that means we get 2 times 2 or 4 ATPs out of this process. And that is done by substrate level phosphorylation, okay? Which is very different from oxidative phosphorylation, which produces the majority of the energy. Okay, so this is a image by BioNinja. Thank you, BioNinja. And it just summarizes it nicely. So we have our 6-carbon compound. We add in our 2 ATPs. That's our phosphorylation. Then just here, that is our lysis phase, where it splits into the 2 3-carbons. Then here we have our oxidation, and you can see that happens twice. So we get our 2 reduced NADs, and you can see that BioNinja writes this as NADH plus H plus. As I said, there it doesn't really matter how you write this. And then we have our ATP formation, and we have 4 of those. Now, if we look at the net gain, we can see we've got a net gain of 2 ATPs because we spent 2, we made 4, so our net gain is 2 ATPs, and our end point of glycolysis is this molecule pyruvate, and obviously we have 2 pyruvate. And the pyruvate, because this is all taking place in the cytoplasm, the pyruvate now diffuses into the mitochondria to crack on with the next stage of respiration, which is the link reaction.